Hi, welcome to my presentation. My name is Lucas Rebelo. I'm a PhD student in the lab of Dr. Chen-Hee Lee in the Department of Animal Sciences at OSU, studying dairy cattle nutrition. Today, I'll be presenting the results of our recent research that explored lysine metabolism in lactating cows. There are two major reasons why we are interested in understanding lysine metabolism in lactating cows. The first is because lysine is the first or second most limiting amino acid for milk protein synthesis. So a correct supply of lysine is critical for milk production. To aggravate this, the major ingredients in a typical dairy cow diet, corn and soybean meal in this case, have relatively low concentration of lysine compared to what is synthesized in milk. In practice, this means that it's not possible to provide enough lysine by feeding corn and soybean meal alone without overfeeding other nutrients, feeding overly expensive diets, or even causing health problems. And the second reason why we are studying lysine metabolism is related to the increasing worldwide demand for improvement in dietary nitrogen utilization efficiency. As this picture shows, the efficiency to which the cow usually convert diet nitrogen into milk nitrogen is low compared to what is excreted in feces and urine. In one hand, if we increase the nitrogen supply, it might result in more nitrogen going into milk protein synthesis, but excretion might increase as well. But if we are able to increase the efficiency to which the dietary nitrogen is utilized for milk protein synthesis, we can consider decreasing the nitrogen concentration in the diet of these animals and still obtain the same milk protein yield. And practically, what we would be doing with this strategy is a reduction in nitrogen waste. And considering the importance of lysine for milk protein synthesis, a better knowledge on its metabolism should allow us to provide enough amino acid for an adequate production, even with a reduction in diet protein. And this ultimately improves the nitrogen utilization efficiency, which, by the way, directly relates to lower feeding costs, since protein is usually an expensive feed and lower nitrogen excretion. And this is made increasingly important by the fact that nitrogen excretion into the environmental is a well-known cause of environmental problems, and eutrophication is just one of them. Therefore, in this study, we hypothesized that a greater lysine supply to dairy cows would increase the amount of lysine utilized for milk protein synthesis, but would also increase the lysine excretion. To test this hypothesis and to better understand lysine metabolism, we evaluated how the increasing supply of lysine affects its utilization, excretion, and contribution to other amino acids. All of these using a stable isotope technique. Although there are several works evaluating amino acid availability through whole body amino acid flux or plasma amino acid appearance rate using a stable isotope technique as well, they were not able to estimate utilization rates of the absorbed amino acid in the small intestine for milk protein synthesis. And that's why our study is unique because with a stable isotope, we were able to measure the duodenally digestible lysine supply and utilization rates of the absorbed lysine in the small intestine for milk protein synthesis. And both measurements are important because utilization rates change according to the supply of amino acid. To perform this study, we used a 4x4 Latin square design with 4 rumen-cannulated lactating Holstein cows with 4 treatments in 4 periods, and each of these 4 periods consisted of 17 days. The first 12 days were used as an adaptation period, while the last 5 days were used as a for abomasal infusion and samplings. All of our treatments were fed a basal diet from the day 1 to the day 17 of each experimental period, and this diet consisted of corn and alfalfa salad with a regular grain mix to meet all the requirements according to the NRC, except that metabolizable protein supply was deficient in 5%. While our control treatment received only the basal diet, our fourth treatment, the RP, received the room protected lysine top dressed on the feed bank from the day 1 to the 17. Meanwhile, our treatments lysine 25 and 50 received continuous abomasal infusions of 25 and 50 grams of lysine per day. To measure the pathway to which lysine was flowing through the cow's body, the abomasal infused all cows with a stable isotope marker, in this case, lysine enriched with 15 nitrogen. All the abomasal infusions happened in the last 5 days of each period. On these pictures, you can see the infusion line that was placed in all cows, passing through the rumen cannula, and our infusion pump setup. With regards to sampling, we performed total collection of feces, urine, and milk, in addition to the collection of blood. Feces, urine, and milk were each analyzed for total nitrogen, and all samples were analyzed for 15 nitrogen enrichment of amino acids. By continuously infusing the marker lysine enriched with 15 nitrogen into the abomination of cows, the idea is to label the lysine pool in the body until it reaches a steady state. And in a steady state condition, differences in 15 nitrogen enrichment should be due to the diet treatment differences. By looking at these graphs, we can see that the enrichment of 15 nitrogen in milk, feces, and urine samples were successful and reached a steady state condition on day 4. Therefore, samples obtained from the day 4 and 5 of the total collections were then utilized for the calculations. We then statistically analyzed the results with a mixed procedure on SAS with cow as random and treatment as fixed effects. Jumping into our results, it's important to note that we did have to exclude data from the control treatment from our final analysis because of problems we encountered with the abomasal infusion. And I wanted to remind you that we infused the marker 15 nitrogen on lysine. Therefore, if other amino acids in plasma and milk were found enriched with 15 nitrogen, it indicates that transamination happened somehow. In regards to plasma amino acid, as you can see in table 2, the 15 nitrogen enrichment occurred in several amino acids, but it was mostly on threonine, alanine, and glutamic acid. Here, it's important to mention that from a biochemical standpoint, lysine does not receive direct transamination, but can be oxidized through other pathways and produce glutamic acid, which is then widely transaminated to produce other amino acid. And this is probably what happened. In contrast to plasma amino acid, as shown in table 3, almost all milk amino acids were observed with 15 nitrogen enrichment. As in blood, the enrichment in lysine was obviously greater because we were infusing lysine enriched with 15 nitrogen, but about 27% of 15 nitrogen was found in other amino acids. In regards to differences in the degree of transamination according to the lysine supply, overall there was not much difference. I would just highlight here the glutamic acid in milk, which increased with the greater lysine supply. And because no oxidation of lysine was previously observed in the liver, this data suggests that peripheral tissues were possibly the responsible for transamination of lysine after oxidation. In regards to lysine metabolism, we observed that 12% of the isotope marker infused was excreted through feces, which indicates that lysine in the small intestine is not 100% digestible, but it could also be due to cycling of 15 nitrogen back to the lower gut. We also found that overall, most of the absorbed lysine was taken up by the mammary glands, while the rest was excreted in urine. When we increased the abomasal lysine supply from 25 to 50 grams per day, we obtained an increase in the absorption of lysine from 132 to 152 grams per day, which led to an increase in the amount of absorbed lysine that was utilized for milk from 108 to 117 grams per day. However, we should note that these results were followed by a numerical greater excretion of nitrogen originating from the greater supply of lysine. Finally, I would like to highlight here the lower efficiency of absorbed lysine that was put into milk for lysine fed as rumen-protected lysine compared to a continuous abomasal infusion of 25 grams of lysine. In addition, feeding rumen-protected lysine tended to increase its excretion in urine when compared to 25 grams of lysine abomasally infused. Therefore, the method of additional lysine supply possibly impacted utilization of lysine after absorption as well. In this case, I'm comparing a continuous abomination supply of lysine versus rumen-protected lysine fed through the diet. Therefore, our results confirmed a large flexibility of the mammary glands to support milk protein synthesis through amino acid transamination, and we were also able to conclude that increasing the digestible lysine increased lysine uptake by the mammary gland, but also increased the lysine oxidation, contributing to other amino acid for milk protein. This transamination should be an important process to support milk protein synthesis by providing amino acids that are in short supply. And lysine likely supports many branched chain amino acids and non-essential amino acid, as shown by our results. And such information is valuable to advance knowledge of lysine metabolism in lactating cows. Specifically, information like this is intended to have a contribution to existing protein supply models, providing further information and prediction of amino acid supply for milk protein synthesis. And we need to remember that good predictions are the baseline for a correct supply of nutrients. An additional step that we would be interested in studying following this study is the investigation of the energy coming from the carbon of the oxidized lysine. In this study, we were tracing nitrogen, but we can think of tracing the carbon from lysine as well, which may further increase our knowledge in the metabolism as a whole. And again, basic studies on metabolism like this are intended to improve profitability and of the production settings and to reduce the waste, which has environmental implications. With that, I would like to thank the partial funding from the sponsor you see here and acknowledge the help of those listed below. Therefore, thank you for your attention.